Welcome everyone. In this chapter, we are going to learn Introduction to Biochemistry. We will understand what biochemistry means, why it is important in pharmacy, and how the cell is organized in a biochemical way. In this session, we will cover two main topics. First, we will study the scope of biochemistry in pharmacy, meaning how biochemistry help in medicine and healthcare. Second, we will study the cell and its biochemical organization, which means what a cell contains and how different parts of the cell support life function. Biochemistry is study of the chemical substance and chemical reaction that happen inside living organism. It uses ideas from chemistry, biological, and physics to understand how life works in biochemistry. We study important biomolecules like protein, nucleic acid, carbohydrate, and lipid. We also study enzyme because enzyme control and speed up most reactions in the body. Biochemistry also explains metabolic pathway and how body processes and are controlled by gene and molecule. Biochemistry is very useful in medicine, agriculture, and biotechnology. Now let us see a short history of biochemistry. Many important discoveries world this subject step by step. The cell theory explains that all living things are made of cells. Later important work in genetics and evolution helped us understand living system better. In 1828, Euler prepared urea in laboratory which showed the organic compound can be produced outside the body. Pasteur's work explained fermentation and the role of enzyme. Later, scientists discovered many metabolic pathways and in 1953, Watson Crick discovered the structure of DNA. All this discovery formed foundation of modern biochemistry. Biochemistry is a subject that connected chemistry and biology. Chemistry explains molecule and reaction. Biology explains living organism. Biochemistry combines both of explain life at a chemical level. It tells us how the body gets energy, how cells grow, how proteins are formed, and how disease happen when this process is wrong. General biochemistry deals with the chemical substance present in the body, such as enzyme, hormone, protein, carbohydrate, fat, DNA, and RNA. In biochemistry, we study where these substances come from, how they are formed, what function they do, and what happens when they are not normal. We also study deficient condition and related symptoms. So general biochemistry helps us understand normal body function as well as disease condition. Now let us understand the aim of biochemistry in pharmacy. Biochemistry is very important for pharmacy because medicine acts inside the body through biochemical reaction. The biochemistry help in pharmacy understand disease, understand laboratory test result, and understand how drug work. It support drug discovery, drug action, side effects, and monitor during treatment. In clinical pharmacy, biochemistry is used to understand and monitor patient condition. For example, blood sugar tests in diabetic, liver function tests, kidney function tests, and lipid profile are all biochemical tests. These tests help in diagnosis and also help to check whether treatment is working properly. So biochemistry support patient care in practical way. Biochemistry is also used in biotechnology. Many modern products like vaccine, diagnostic kit, and biological medicine are developed using biochemistry knowledge. Also, biochemistry support pharmaceutical formulation by improving drug stability, drug compatibility, and better perform inside the body. It also helps in identifying useful natural compounds from plant and microbe. Now we move to the second major topic, which is cell and biochemical organization. The cell is the basic unit of life. Inside cell, many chemical reactions happen continuously. Each part of the cell has a special function. Understanding the cell help us understand how the body works at the molecular level. This slide shows the molecular composition of cell. The cell mainly contains water, organic compound, and inorganic compound. Water forms about 70 to 75% of the cell. Organic compound include protein, carbohydrate, lipid, nucleic acid, amino acid, hormones, vitamins, and coenzymes. Inorganic compounds include minerals and iron such as sodium, potassium, calcium, magnesium, chloride, phosphate, bicarbonate, and others. All these compounds together support normal cell function. Now we study the structure of nucleus. The nucleus is present is eukaryotic cell and it is the control center of the cell. The nucleus contains DNA which carries genetic information. The nucleus is surrounded by a nuclear membrane and inside it there is nucleoplasm and nucleus. The nucleus controls cell activity by controlling gene expression and protein production. Now let us understand nucleus. The nucleus is a small dense body inside the nucleus. Its main function is to form ribosome. The ribosome are essential for protein synthesis. So nucleus is important because without ribosome the cell cannot produce proteins and without proteins cell function cannot continue. Now we see cytoplasm and cytosol. The cytoplasm is the area outside the nucleus where most organelles are present. The cytosol is the liquid part of cytoplasm. Cytosol contains many enzymes and is the place for many important reactions. Some pathways like glycolysis and other metabolic reactions occur in the cytosol. So cytosol is an important area for biochemical activity. Now we study the structure of mitochondria. Mitochondria are organelles present in the cytoplasm. They have an outer membrane and an inner membrane. The inner membrane form fold called cristae. Inside mitochondria there is matrix. The structure is designed to support energy production. Mitochondria are called the powerhouse of the cell because they produce ATP, which is the energy currency of this body. Important processes like the electron transport chain and oxidative phosphorylation happen in mitochondria. Many energy-related metabolic reactions occur here. Cells that need more energy, like muscle cells, have more mitochondria. Now we learn about lysosome. Lysosomes are small vesicles present in the cytoplasm and surrounded by membrane. They contain digestive enzymes, so lysosomes are sometimes called the suicidal bag of the cell. This enzyme work based in acidic pH. Lysosome digests material taken inside the cell. The processing like phagocytosis cells like macrophages contain many lysosomes because they digest game and harmful substances. Now we study peroxisome. Peroxisome are small membrane-bound organelles involved in several metabolic reactions like oxidative reaction. They take part in lipid metabolism and breakdown of substances like D-amino acid and polyamine and like bile acid. During this reaction, peroxidase may be produced. Peroxisome contains enzymes like catalase and peroxidase which convert harmful peroxide into water and protect the cell. In plant, peroxisome also support photosynthesis and seed germination. This slide shows a diagram related to the internal membrane system of cell. In the cell membrane form an important network for processing and transport of protein and lipid. The endoplasm reticulum and Golgi apparatus are part of system. This organelle help in producing, modifying and sending protein to the correct location inside or outside the cell. Now we study the endoplasm reticulum. Endoplasm reticulum is continuous with the outer nuclear membrane. It looks crushed because ribosomes are attached on the surface. The membrane enclosed canal are called cisternae. Ribosome and endoplasm reticulum are mainly involved in protein synthesis. The protein synthesis produced and enter the cisternae and later move to the plates or further process and secretion. An extensive network of membrane in the cytoplasm called cytomembrane. The cytomembrane includes the endoplasm reticulum and Golgi apparatus. The endoplasm reticulum of two types, crushed endoplasm reticulum and smooth endoplasm reticulum. This whole system helps in making protein and making lipid and transporting substance in the cell. Now we study the Golgi apparatus. The shape of Golgi apparatus can change depending on the cell type and functional stage. In some cells, it looks like a network, and in some, it looks like chalk or blood. In nerve cells, it is seen as a network surrounding the nucleus. These organelles play an important role in processing and packing. The main function of Golgi apparatus is packing and secretion of protein, which receive protein from endoplasmic reticulum and modify them. Golgi apparatus help to some other organs like lysosome and parasitome. It also help in protein targeting, meaning it sends protein correct location, including the cell membrane. It performs processes like glycosylation and sulfonation of protein. It's also the site of synthesis of some lipid like glycolipid and picomaline. In some cases of diabetes, diffusivity process in insulin in Golgi complex is involved. Now we study the cytokeratin. Cytokeratin gives shape and support to the cell and also help in movement and transport inside the cell. Cytokeratin is made of different protein structures such as microtubules, microfilament and intermediate filament. These structures act like the internal framework of the cell. Microtubules are made up of tubulin protein and help in cell movement and transport. Microfilaments are made of acting like protein and help in the shape and movement. Intermediate filaments are stronger supporting fiber and provide stability. Examples include keratin skin and neurofilament in nerve cell. So, cytokeratin help in the remain stability functional. This is transport process across the cell membrane. The cell membrane controls what enter and leave the cell. Transport can be classified mainly into passive diffusion, facilitated diffusion, and active transport. Each type has own method depending on the type of substance transported. Passive diffusion is a simple process where molecules move from higher concentration to lower concentration. Does not need energy. Small molecules like oxygen and carbon dioxide move through the membrane by passive diffusion. This process depends on concentration difference. Facilitated diffusion is also movement from higher to lower concentration, so it also does not need energy. But here the transport needs the help of the special carrier protein in the membrane. For example, glucose and some amino acid enter the cell using carrier proteins. Active transport occurs against the concentration gradient, meaning from lower concentration to higher concentration. This needs energy, usually in the form of ATP. Ion pumps like the sodium potassium pump are example of active transport. Active transport is very important to maintain proper ion balance in the cell. Finally, transport of large macromolecules such as protein, polysaccharide and polynucleotide across membrane is also important. happen mainly two mechanisms, endocytosis and astrocytosis. Endocytosis means intake of macromolecule into the cell. For example, uptake of LTL by cell. Astrocytosis means release of macromolecule from the cell. For example, secretion of hormones like insulin and PTH. This process helps cells communicate and function properly.